Hello, my name is Elaine and my partner is Anya, and today we're going to be talking about CRISPR. So the contents that we will be discussing today is the intro of CRISPR, the origin of CRISPR, the uses and how it works, gene therapy versus engineering, case 1 and case 2.
Okay, so what exactly is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it is a DNA cutting protein. It was first discovered in archaea and later in bacterial immune systems. In bacteria, it helps them defend themselves from attacking viruses. The CRISPR enzyme cuts off pieces of viral DNA and it combines this viral DNA with its genome in between specific spacer regions. And by storing the viral DNA, the bacteria remembers this virus and is able to counterattack it if it invades again. So there are two components of CRISPR. There's Cas9 enzyme and the guide RNA. The Cas9 attaches itself to a common sequence in the genome/PAM using guide RNA. Um, and it attaches at a protospacer adjacent motif, which is the site at which the Cas9 enzyme is to cut or cleave the DNA at. So the guide RNA unwinds part of the double helix and binds with one half, and the Cas9 will cut this area. If the bacteria tries to fix this error, there is a possibility for mutations to occur, making CRISPR a good tool for disabling a gene.
So now that we know all of those facts about CRISPR, how was it discovered? It was discovered by Jennifer Doudna, a Nobel laureate in chemistry, and Emmanuel Carpentier. Their research focuses on RNA, where Cas9 was then discovered in their lab's research into RNA biology. What are the uses of CRISPR? CRISPR-Cas tools are being used to correct genetic variants with the hopes of treating many human genetic diseases like inherited blood disorders, such as sickle cell disease, inherited eye diseases like retinal degeneration, muscular genetic diseases, genetic liver disease, congenital genetic lung diseases like cystic fibrosis, neurological disorders such as Parkinson's, Alzheimer's, and Huntington's disease, and many more. CRISPR systems have been utilized to treat infectious diseases such as viral infections like HIV, hepatitis virus infections, and more. CRISPR-Cas12 and CRISPR-Cas13 systems have been recently used to inhibit HIV infection. Studies have applied CRISPR-Cas systems for effectively targeting different genes and have managed to prove the potential treatment ability for initiation or progression of lung cancer, breast cancer, and many more. CRISPR-Cas9 could inhibit multiple steps of HIV infection. In fact, several research labs have made excellent efforts to improve the use of CRISPR-Cas9 for treating HIV infection. Now that we know the use of CRISPR, let's move on to how it works. The mechanisms of CRISPR-Cas9 genome editing can be generally divided into 3 steps: recognition, cleavage, and repair. In the recognition stage of CRISPR system, the CRISPR system locates the DNA it is programmed to find. The designated sgRNA, which is a guide RNA designed for guiding the Cas9 protein or other Cas proteins to cut DNA. It directs Cas9 and recognizes the target sequence and the gene of interest through its complementary base pair component. Then it binds to DNA. This means that the guide RNA recognizes the sequence of DNA to be edited and then binds onto the sequence. During the cleavage stages, the Cas9 cuts the DNA strand in half. Lastly, in the repair stage, the initiation of desired mutations, sequence insertions, or gene deletion proceeds. Now, before we get into the next information, we need to understand what DNA is first. DNA consists of genetic codes with sets of rules. The genetic code consists of 4-letter codes known as 4 nucleotide bases of DNA. This includes adenine, which is A, cytosine, which is C, guanine, which is G, and thymine, which is T. These codes have specific pairs of C and G and A and T. However, RNA consists of the same codes as DNA, except thymine is switched with uracil, where A bonds with U and C bonds with G. Now that we learned the basics of DNA and RNA, we can now move on to the next topic: steps to use CRISPR-Cas9. To use CRISPR-Cas9, Scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular code. The guide RNA is then attached to the DNA cutting enzyme Cas9. It is then introduced to the target cells where it locates the target letter sequence and cuts the DNA. Scientists can then edit the genome by modifying, deleting, or inserting new sequences.
So, to further tell what other uses that CRISPR can be used for, there's both genetic engineering and genetic therapy. So, genetic engineering is when scientists use recombinant DNA, which is when is created from two or more fragments from different sources. They use these techniques to introduce new characteristics or traits. And then with genetic therapy, the genes of a genetic variant are either replaced or modified to restore or enhance cellular function or to improve responses to non-genetic therapies like chemotherapy. What they both have in common is their goal, which is to alleviate human pain and suffering of a disease. However, with manipulating genes, those who only look to accumulate desirable traits or to improve their traits have given rise to multiple ethical debates. There are some dangers involving these processes, and this is because genetic alterations can be risky to both the individual undergoing the process as well as their offspring. With somatic cell gene therapy, which targets the body cells rather than the germ cells, future generations are safe. However, with germline therapy, where the genome of the human gametes are targeted, which are the sex cells, the offspring may receive these alterations. Hence, there may be unpredictable and irreversible side effects. So before I talk about what Casgevy is, which is the first case of what we're going to be talking about with CRISPR, what exactly is sickle cell anemia? So sickle cell anemia is a hereditary disease that is mainly found in African Americans. It is caused by a mutation in hemoglobin protein found in red blood cells, which are used to deliver oxygen to the body's tissues. This mutation causes red blood cells to become hard, brittle, and sickle cell shaped. Cells get stuck within vessels and scratches them, leading to inflammation and limited oxygen delivery. Those with this disease have chronic pain, anemia, fatigue, and a shortened life expectancy. Prior to Casgevy, there were and still are medications and blood transfusions that can lessen the pain, but this is only a temporary fix because it is not sufficient for some. There's also bone marrow transplants, but patients with this disease can't undergo a bone marrow transplant because they need full and healthy siblings. Only about 15% of patients have a matched sibling, and unrelated donors are typically found for only 10 to 12% of these patients, meaning that only 25% are able to get the bone marrow transplant. However, this all changed when Casgevy was approved on December 8, 2023 for those 12 years and up. So there are different versions of hemoglobin in the body. Fetal hemoglobin is produced during fetal development and is most effective at carrying oxygen, but it is sometimes turned off after birth and replaced by its adult version, which can be affected or caused by SCA-causing mutations. This change is due to the protein BC11A, which tells the body to reduce fetal hemoglobin production and make more of the adult hemoglobin. Using CRISPR-Cas9, scientists in a lab increase fetal hemoglobin production by modifying the gene that encodes BC11A and reduces activity. This lowers the production of BC11A, enabling more fetal hemoglobin to be generated and preventing red blood cells from sickling. Prior to this treatment, the patient must undergo intensive high-dose chemotherapy, also known as myeloablative conditioning, to remove the patient's hematopoietic stem cells in the bone marrow, and these are stem cells that can differentiate into different blood cells like red blood cells and white blood cells. Then these newly edited cells are then injected back into the patient via a stem cell transplant. So for case two, this revolves around Alzheimer's, and Alzheimer's disease is the most common neurodegenerative disease, and it is caused by an accumulation of tau and amyloid proteins, which can cause neurofibrillary tangles and inhibit communications between the neurons. An accurate and proper diagnosis of AD can be made after death, but there are still non-invasive diagnosis options being experimented with and developed, like the mini mental state examination and its more detailed alternative, the St. Louis university mental statics exam. So AD can be both sporadic or occur through dominant autosomal mutations, meaning that they get passed from parent to offspring through the 22 chromosomes that are not the sex chromosomes. When it is hereditary, three genes are related to the cause of this disease: the amyloid precursor protein, which makes amyloid, and presenilin 1 and 2, which hinders the ability for the body to digest the amyloid. So some doctors, before using CRISPR, they used mice, um, which is the most common animal used in uh, gene therapies. So they prepared this mice by simulating Alzheimer's disease, and the majority of them were generated by overexpressing human mutated genes by having transgenic mice. And this meant that they had AB and tau proteins, a lot of them creating neurofibrillary tangles, like I mentioned before, and this allowed scientists to better understand some aspects of AD pathogenesis, but the mouse models did not represent an accurate neurodegeneration. So Barty Strooper, he was a molecular biologist, and he's based in KU Leuven in Belgium. He and his group used CRISPR to make mouse and rat models of AD, and they altered the APP gene in mice so it would represent the version of the gene found in AD human patients. And this was done through introducing 3-point mutations: the G676R, F681Y, and R684H. And in doing so, they generated humanized animal models that will be useful for studying APP processing and unveiling new mechanisms of disease pathogenesis. Now, using this method, another scientist, Daniel C.S. Tan, and his colleagues used CRISPR-Cas9 to introduce a short deletion in the transcriptional start codon in exon 1 of the MAP gene, which is the gene that creates tau proteins. This mice did not have any clear phenotype, but compared to previous mice, they were less probable to excitotoxic seizures, which is when your brain is overstimulated, and they did not show any memory deficit. Thank you.
Thank you. That will be it for today's presentation.
Yeah,
And
thank you for this presentation. Oh, sorry, cut you off. Go ahead.
sorry, here's the sources that we used. Yeah, that's it.
Okay, yeah, thank you so much for the presentation, Elaine and Anya. I think CRISPR is kind of like a new frontier in the field of medicine and research. I think there's a lot of possibility to find new treatments for different diseases as presented in the different cases that Elaine and Anya showed us. I think while addressing those possibilities, it's also important to talk about like the dangers and the ethics of this new technology. And I think this presentation addressed like both aspects. So I really appreciated that. I wanted to clarify in some of the slides, there was like a mutation, and it would be like a letter followed by a number and then like another letter. So you might see that a lot in the future. Let me try to point out. Okay, so in one of the slides there was G676R. So the letters stand for the different amino acids. There are 20 amino acids used by our bodies, and then the number in between the letters stands for the position of that amino acid. And so the first letter is kind of what the wild type or the original sequence has. And the second one is the second letter at the end is what it's changed to. So different amino acids have different properties. Some of them are acidic, some of them are basic, some of them are positively charged, some of them are negatively charged, and some of them are uncharged. And so when you change the amino acid, sometimes if you change it to an amino acid with like a similar property, it might not cause big changes. But if you change like a charged amino acid to one that's not charged, that might affect like how the protein is folding, and that ultimately affects the function of the protein. And as you'll learn in future courses, there are different types of mutations based on changes in like nucleotides, and some of them can translate to changes in amino acids, and some of them, because there are so many codons that code for similar amino acids, it might not cause differences in amino acids. And this technology is obviously very powerful, and it's exciting to see where it might go in the future.